like the previous presenter, thank you very much for staying here and avoiding the bar. I think it's a testament to all of you. And I also want to thank the organizers and people on the board of ARM for letting us substitute for the presentation that was supposed to be scheduled. We really do appreciate the opportunity. So today I'm going to talk to you about our closed system approach for reducing, significantly reducing, I should say, variability, the amount of time, the cost, and the labor required in manufacturing CAR T cells. What we're using is a cell-sized version of a Pachenko. For those of you who don't know, if you think about going back to a coin sorter with nickels, dimes, and quarters, this is essentially a miniaturized version or a micron-sized version of a coin sorter. The technology was originally developed by Princeton, and we've licensed it exclusively and have transitioned it from a deep silicon device into a polymer-based device. And it's taken us some time, but we are there. And the benefits of this technology is you really get very, very gentle and uniform processing of cells on the basis of size. So cells can be fractionated, so blood can be fractionated based upon size cutoffs. And to use this, basically, we just add a very low pressure, simple pressure, about 10 psi, and we push blood through the device, and the cells that are bigger than the gap sizing that are shown in that diagram there bump in towards a central channel to be collected. It's as simple as that. There are no moving parts. It's a very passive device, and it's very, very efficient. And I'll show you how efficient in the next few slides. But what it enables for cell therapy and cell therapy manufacturing in particular is a nice closed system that effectively separates, it washes, and it concentrates all at the same time. And so we can manage cells throughout the continuum of cell manufacturing processes. As I'm sure many of you are aware, there are lots of challenges to scale up of CAR T cells and other cell manufacturing processes. A lot of discussion at this meeting in particular about the challenges of logistics, especially this morning, around what are the real issues in growing the business and manufacturing and scaling up to meet when CAR T cell therapies become available for solid tumors, for example. So donor variability is an obstacle. There's no doubt about it. There's a component of donor variability. But I'm going to show you today that donor variability is not as big as you think it is. A lot of it is actually coming out of the process. Typical cancer patients obviously are immunosuppressed and have low white counts and T cell counts, so every cell that you lose is critical. There are often some significant flaws in the current process when you look at it from a process manufacturing perspective. We have re-stimulations, often because the donor is not responsive. We have second attempts at transductions because the virus system isn't as efficient as it could be, or we've lost too many cells. Current processing solutions realistically have been designed many, many years ago, and they've been designed for blood banking. They've been designed for handling cells that are relatively available. They're coming out of an apheresis product, and if you really need a few more cells, you have the patient sit there for a little bit longer, another 15 minutes, and get the more cells, and you're really not very concerned about how much cells you're going to lose. Contrast that to CAR T cells. You've modified the cell with a very expensive construct, and you really don't want to lose any more of those cells, and the patient needs those cells desperately. In addition, a lot of people are still using FICOL. This is an open system. Some people are using it in centrifuges. Some people are using it in closed systems. But the point of the matter is FICOL is not the best solution, and I'm going to show you why. In addition, people are cryopreserving samples. It's a cardinal rule of cell handling to not do harm. When 10% of your T cells are going to be damaged by a cryopreservation process, it makes no sense to us to lose 10% of a precious cell from a depleted patient in the first place. So why start out 2 yards behind the start line? And so then you have the washes that people think about, the losses of 15, 20% associated with automated cell washing. This is a compounding problem, as you all know. So you're going to cryopreserve it. You're going to wash it out of cryopreservation media when you get it into the central processing facility. You're going to put it back into culture, and then eventually you're going to start activating the cells. this process is inherently highly variable. There's a significant amount of skill required on all the operators involved. There's going to be stress and pressure as this industry tries to grow in how do you get enough skilled technicians to actually perform these manual tasks. And with that manual task, there's a high risk of error. And obviously, with a high skilled labor, there's an even higher cost. And at the end of the day, you're starting the process of transduction or transfection with a 50% cell loss of what you actually harvested from the donor. And you've lost 3 to 5 days as far as the patient's concerned. So I'm sure many of you in the audience are familiar with the general workflows around CAR T-cell manufacture, so I'm not going to belabor the point too much. But what we're trying to do at GPB is develop a solution for a point of collection apheresis to T-cell in culture media solution and deliver cells in culture inside 3 hours. Okay? We're going to do that by processing through our microfluidic chip at a rate of about 5 times 10 to the 10th, so a half liter in less than an hour. And we're going to have an overall process with isolation and activation of the T cells and returning them into culture inside the 3-hour window. With that, we'll have exceptionally efficient removal of red cells and platelets, and we'll give back to the transfection process or transduction process 90% of cells that were there in the patient's sample as compared to about 50% if you're comparing against FICOL. We will eliminate the cryopreservation and all of those associated cell losses, and you get activation on a day of apheresis. And so that gives you a 3 to 5 day reduction by the time you've taken in the logistics, the handling, the preparing, the getting the cells happy to be activated and expanded before you can actually put virus onto the cells. So to give you a sense of what the product looks like, the cell product, on the left you can see labeled waste and product. So we have leukocytes clearly in the white cell, and you can see there's very, very few red cells, essentially zero. We have more than 99% red cell elimination. In terms of platelet contamination, we're removing significantly greater than 90%. We're still trying to fine-tune that part of it. And what we've been doing to stress test the system has been using apheresis products from an LRS fraction, so exceptionally high platelet counts, 3,000 per microliter, as well as high white counts of around 150 to 160 million white cells per mil. And we're capable of processing that with no problem. Okay? If you compare and contrast on the three micrographs there, the green is where platelets should be on the DLD product side, and on the first wash, there's still quite a few platelets in the FICOL. On the second wash, there's still platelets in the FICOL. And this is because they're aggregated and they come down and level out on the top of the FICOL interface. So FICOL really does not allow the removal of platelet aggregates. And anytime you do an apheresis, platelets get activated, and this is what happens, and especially when you start shipping it overnight. So this is a fairly dense slide, and I apologize. So I'm going to go through block by block. The top two blocks are our cell recovery. We get 99.5% of the cells going in, coming right back out of a device. Okay? This is not a typo. Similarly, on PBMCs, accordingly, you would expect a similar recovery, and we get that at 99.6. What we did here was we compared those against an ammonium chloride lyse, did 2 washes, 1 or 2. You can see the gradual losses with an ammonium chloride lyse, which a lot of people do use, and then compared it to a FICOL prep. This is from 24-hour-old apheresis products. And the FICOL range is anything from 25% to 60% recovery, with about 50% being the average recovery after FICOL. And we've looked at many donors. But when we take cells from this process and we stimulate them, activate them, what we've done here is we took a sample, divided it into 3 ways. We looked at the direct magnetic, the diurnal beads, if you will, and we depleted T cells and put those into culture. We did a FICOL prep and exposed them to the same diurnal beads at the same time, same number of cells, normalized to a few million per mil, so it's optimal conditions for concentration-wise for our T cell expansion. And we did the same thing with the GPB-produced T cells. And what we see at 15 days, following a simple IL-2 induction on day 3 with 200 units per mil, IL-2, is we get twice as many central memory cells being produced using the DLD product as compared to the Ficol and the direct magnet. And going into that reaction, we have twice as many cells coming off the DLD device in the first place. So we have about 4-fold more cells at the point at which you would be looking at activated cells at day 15. If you look at the phenotype conversion, and we look at the statistics about the consistency of response on these cells, the number of donors converted who are making central memory cells with IL-2 induction, I used an arbitrary 50% cutoff here, but only a third of the direct magnet samples were actually above a 50% component or composition of being central memory cells. The Ficol was about 50%. In contrast, every single one of the DLD samples was a central memory conversion, above 50%. If you look at the percentage of the central memory cells in the culture, only half of the cells are in for Ficol or for the direct magnetic fraction. In contrast, the GPB product has 75% central memory cells. But the most impressive thing about this, and why it's really important for how you think about starting a manufacturing process, is to have something that is reproducible. And the CVs of the DLD-produced process, the consistency of response, show very clearly at 13% CV, but the large majority of the bias or the heterogeneity that is in current CAR T-cell manufacturing is actually coming from the process and not coming from the biology. Now this, I think, is why we're really excited about the future for this technology. And I want to point out that concentration steps, washings of media, can also be achieved throughout this whole continuum of workflow using a DLD technology. So I just think we have something that's really very exciting and very promising. We're very fortunate. We get to work with some of the best in the business. We have Kurt Sibben, the founder of the CD34 antigen and the pioneer of stem cells. And from a technical side, we have Bob Austin's group out of Princeton with Jim Sturm, who are just phenomenal and brilliant people capable of helping us accelerate how fast we can process these samples even more. And working with Nancy Hardy, who was at the NIH, is now at the University of Maryland at the Greenbaum Cancer Center, who is helping us get access to patient samples as we go forward. And my old mentor from Becton Dickinson, where I used to work, Dieter Rechtenwald, who has had a lot of experience working with Miltenyi in his former life after he left Becton Dickinson. So I have a strong group of people supporting us. And with that, I want to kind of summarize where we are and what we think we've got here. So we have a closed system. We're going to be able to reduce variability. I hope I've demonstrated that to you. It's just a striking finding, not only at a process level from a cell counts and cell variability, but also at a biology level. And we're going to be able to reduce cell losses, improve the variability. We're going to remove, by putting this into one system solution, all the error-prone steps and a lot of the handling steps. And by doing this, we'll reduce the overall time that the patient is waiting for a drug, which is really what drives us. And bystander effects, if you will, benefits for businesses are significant. Manufacturing processes now can be reliable and reproducible. The number of relevant cells going into culture compared to other approaches is going to be much higher. And finally, the capacity of central processing facilities won't have to be as significantly invested. This given square footage or footprint, if we use this solution, will dramatically improve the scheduling and improve the capacity, therefore, because you don't have to have all the extra equipment. With that, I'd like to say thank you very much. And I'll take any questions outside or right now if you have any. Thank you.